Welcome to Generation Hope, the interview series. I'm your host, Ron Clayman. Today, I have the distinct honor and pleasure of introducing Monica Coenraads, CEO and founder of the Rett Syndrome Research Trust, a group laser focused on curing Rett. Under Monica's leadership, RSRT has raised over $123 million to drive genetic medicine research and translation. RSRT is the largest funder of Rett research worldwide and is advancing genetic medicines that target the root cause of the disorder. The gene therapy programs, now in clinical trials, were started and incubated at RSRT, and the trust has become a go-to partner for scientists and companies in genetic medicine. Monica's impact has been recognized around the world. She holds honorary doctorates from UMass Medical School and the University of Edinburgh, and she's a recipient of Redbooks' Mothers and Shakers Award. Most recently, she was named to Forbes 2025 50 over 50s Impact List. This conversation was recorded in October of 2024. Stay tuned after the interview for an update on the Neurogene and Taysha gene therapy trials, plus updates on other key initiatives underway at RSRT. And now, Monica Coenraads. When you tell your story, where does it begin?
Well, if you want to hear my story from the beginning, I guess it's when Chelsea was born. Mm-hmm. She was born in 1996. She was our first child, and we had been married 9 years. First grandchild on both sides of the family, so eagerly awaited baby. The first year was wonderful. It was the first time I was actually home. I had been running a family restaurant, which we had sold right before I got pregnant. And we had waited, I was working crazy hours, 100 hours at the restaurant a week. So we had waited for to sell the restaurant before we started a family. And the first year was great. And after that, we started noticing signs that, something strange was going on. So it started with, she stopped looking at us. She had kind of this absent look about her. She stopped responding to her name. She wasn't learning anything new. She wasn't crawling. She couldn't get herself into a seated position. She was starting to drop everything. She couldn't hold on to anything. And things got worse from there. So we started trying to figure out what was happening. The doctors didn't know. We did a bunch of testing. We started off with developmental pediatricians, geneticists, neurologists. And then kind of in a serendipity kind of way, someone that I knew through a mom's group that I belonged to, we had all had children the month of October, 1996. She said to me, my neighbor has a child with Rett syndrome. Why don't you look it up? And I went to a website, looked at all the symptoms, and was making these mental, yep. And the next morning, I went to her pediatrician, showed up with no appointment, 7 o'clock in the morning, showed up at her office, waited for her to come. I said, I think Chelsea has Rett syndrome. She's like, I think I heard about it in medical school. I'm not sure. She pulled down a neurology textbook. And anyway, no one thought she had it. This was before the causative gene mutations had been identified. So there was no blood test. Mm-hmm. And we were living in Virginia at the time. We went to Johns Hopkins. They had a Rett syndrome clinic. They weren't used to seeing children that young. She was 20 months old. Mm-hmm. They didn't think she had it either. They said, come back in 6 months. I was like, how the hell am I going to get through the next 6 months? I need to know what's happening to my child. I think I made it to 3 months, and then I made another appointment. And at that point, she had lost more skills, and they diagnosed her tentatively with Rett syndrome. A year later, the gene was found. At that point, I had already sent blood samples to all the labs that were part of this group of doctors that were looking for the cause of Rett syndrome. Mm-hmm. So in October of 99, the paper came out in Nature Genetics that the cause of Rett was MECP2. And within like 2 or 3 days, I got a call from one of the doctors saying, we found a mutation for Chelsea. Yeah, I mean, that's where my story starts. And it getting a diagnosis gave me some peace. Like I knew what it was. And maybe I remember coming home, my mother-in-law was visiting, and I remember telling my mother-in-law and my husband, because I'd had like a frantic month of just trying to figure out what was going on, and, phone calls to doctors, visits, running around here and there. And I remember saying, okay, this is what she has, and I'll accept it. And that lasted about a week. I was like, okay. By the time she had her diagnosis, I had kind of already grieved. Like, before we knew what was happening, I realized that whatever it was going to be very, very serious. And, there was a few days there, maybe a week, where I couldn't breathe. Like, it was tough to get out of bed in the morning. Like, it was, I felt like I had a cement slab on my chest, just this, it was horrible. But it didn't last that long. I went from why me, why my family, why my daughter, right, to why not, let's do something about it. By the time the diagnosis came around, the formal diagnosis with the genetics test, that was almost a relief. Like, I thought it was Rett. Now we know for sure. And then kind of, the next 25 years.
So that spurred you into starting the foundation, because I'm guessing there was nothing at that time.
There was an organization. There was the International Rett Syndrome Association. They were more of a family support group. I was really, really focused on research. And so I've started 2 organizations. In 99, I started the Rett Syndrome Research Foundation. And I was there until 2007. So 7 or 8 years. And, we did what many disease groups do. We started fundraising, and there was very little research being done in the field. So we started kind of fertilizing the field, so to speak, giving small grants, $50,000, $100,000 grants to lots of scientists in lots of different areas, just trying to learn about the disease. So genetics and neurobiology and molecular genetics and all, just lots of different fields. And then in 2007, there was a very important paper that came out from Adrian Bird. He's a scientist at the University of Edinburgh. And he had we were working closely with him. He was the chairman of our scientific advisory board. He had discovered MECP2 in the early 90s. It wasn't until 99 that we knew that mutations in the gene that he had discovered and named caused Rett syndrome. And then it was just kind of natural for him to, now he had a disease that was associated with the gene that he discovered. So, but he came to me 2003 or 2004 with an idea of an experiment, and asked for funding. He wanted to make a conditional mouse model, which means, can we make a mouse that has no MECP2, or the MECP2 gene is shut down, and then at will, you turn it back on by giving the mouse a drug that does a whole bunch of stuff that basically turns the gene back on. It's not something we can do in our children. It was a proof of concept experiment. The mouse is engineered in a certain way to respond to this drug, so that the gene turns back on. And it was a pretty far-fetched experiment for a brain disease. And he's not a neuroscientist and was thinking about Rett syndrome, I think, in a very different way than most neuroscientists would. And so he did the experiment, and it was a long, complicated experiment that had its ups and downs. And but the end result was pretty fabulous. And that's that if you restore MECP2, regardless of the age of the mouse, you could do it in a newborn mouse, a young mouse, an old mouse, a very old mouse, put the protein back, and the symptoms go away. That really stunned the community. It was not a result that anyone expected, including Adrian. It was a result that I expected. And it, you know, I mean, it was great to have it confirmed, but maybe as a parent, I just needed to believe that, you know, I needed to have hope. And it's like, of course the symptoms are going to go away. But finally we had, you know, kind of proof, right? So it was a very big moment, and people still talk about that experiment. And now lots of diseases have done the very same experiment, you know, the bird experiment.
Knockout model.
For you know, for their own diseases. And so at that moment, that really changed everything for me. And I really wanted to focus on genetic medicines. You know, there was a lot of, you know, we have a single gene disease, which a lot of disorders don't, right? Alzheimer's, Parkinson's, autism, schizophrenia. Those are all really complicated, you know, genetic disorders. But Rett syndrome is monogenic, one gene. So we know our target.
Yeah.
It's not degenerative, so brain cells aren't dying. They're all there. They're all in the right place. And now because of Adrian, we knew that if we could restore levels of the protein, the symptoms go away, at least in mice. And so for me, it was just, you know, a no-brainer that we're going to go after genetic medicines. Now, I had a recent conversation with Adrian, and he said to me, you know, the decision to go after genetic medicines was really, what's the term he used? Non-conventional. Yeah. The decision to go after genetic medicines was really non-conventional. You know, everybody's doing small molecule drugs. And because this was, you know, 2008. Yeah. 2009, you know, we funded our first gene therapy work in 2010. And it kind of caught me by, you know, it was kind of a surprising statement. Really? Like, because to me, but that's because I'm not a scientist, I think, you know, and I'm coming after, I'm looking at it through a very different lens, and sometimes a naive lens, right? Of a parent, not a scientist with all the baggage and the scars and the, from
the blinders.
Yeah, and so I was like, of course we're going to go after genetic medicines. Well, now, you know, now everybody's doing it, right? I think the difference with RSRT is that we decided to do that in 2008 when it was still kind of science fiction, but we've stuck with it, and you know, it's exciting to see the progress.
So in 2018, I attended my first conference for Phelan-McDermid syndrome. It's a big conference with families and researchers. And at that time, I think we had Evie's diagnosis for about a year and a half or something like that. And I'd read up on, you know, CRISPR kind of was just kind of being talked about for about a year earlier or so. And they just kind of found it. And I'd asked the question, you know, hey, what about gene therapy? Because nobody was talking about, you know, they were talking also about kind of, you know, therapeutics, you know, medicines and things that to, you know, maybe we can, if we attack it from here, maybe. So I asked about gene therapy, and everyone looked at me like I was, you know, patting me, okay, son, it's okay. You know, maybe 20, 30 years from now. And I couldn't believe that response. And 8 years later, human trials. And I talked to those same researchers, and they're all just like, yep, I can't believe it. None of us, everyone is just so happy and so surprised. But it's like you said, it's like they're operating in their kind of silos. And it's amazing to me, and I'm sure you as well, because you speak to so many different researchers, researching so many different things, that your hub of knowledge is so much greater than each of the individual researchers, because they know what they know. They don't have time to go and learn about all the different research, all the different modalities that are out there. So I think it's really important that, you know, people like you, like parents, you know, ask those naive questions and kind of push for it. I'll tell you a story later about the ICONS conference as well, with all the researchers talking about newborn screening, and I'm still amazed at how removed they are from patients. Yeah. And there needs to be a closer collaboration there. So let's talk about Rett syndrome. Tell me, what is Rett syndrome?
So Rett syndrome is a single gene neurodevelopmental disorder that affects girls primarily. There are boys that have Rett syndrome as well, but it's more rare. And that's because of the genetics, which probably don't need to get into here. But children are born seemingly healthy. They have a fairly typical first year of life. And then somewhere around, a year to a year and a half, they have a regression. They lose skills. It can be a very dramatic regression, or it can be a very subtle regression where the kids really haven't gained that many skills. And it's even difficult to recognize whether they've had a regression. But they typically lose, whatever words they've gained, their ability to use their hands. Typically, if they walk, they can maintain that walking ability for many years, but about half of the children never learn to walk. As the disorder progresses, it's not neurodegenerative, but it is progressive. My daughter is worse at 28 than she was at 20 or 15 or 10. And so there's more symptoms that appear. Seizures are very common, irregular breathing patterns, breath holding and hyperventilation. There's a lot of gastrointestinal issues, constipation, reflux, scoliosis, hip issues. So it's a lot of symptoms. It's a big, long laundry list of symptoms: anxiety, sleep problems. But the girls, I like to say that the children, boys too, they're just so much more than their laundry list of symptoms, right?
Of course.
They've got personalities. They're affectionate. They can learn. So it's a very debilitating disorder. The children grow up to be adults and can live, well into middle age. And
Wendy told me about, in 1 week, she diagnosed a 3-week-old.
Oh, wow.
And a 72-year-old.
Yeah. Wow. What a spread.
What a spread.
Yeah.
So I was telling you, Wendy had some news to share with you. What's the current kind of guesstimation at the prevalence?
Well, we actually don't have good data on prevalence. There's been a few very spotty, studies done, but most people say 1 in 10,000 females, 10 to 15,000 females, which means around 15,000 here in the U.S., 350,000 worldwide. Right. But that's a real guesstimate. And what does Wendy think it is?
All right. Hit play.
Because numbers aren't, they don't have the numbers because most people don't get a genetic diagnosis.
Right.
So with Rett, I'm guessing the numbers are significantly higher than what's published.
And so Rett syndrome is certainly the number that we're seeing, which is still relatively small numbers, but about 1 in 1,500 girls, which is remarkably higher than what has been published.
Six or who's 1 in 10,000?
1 in 10,000.
Yeah,
This is what's been published.
Wow.
That's huge. Right. That's huge. So with that comes more eyeballs on Rett syndrome, more investment into Rett syndrome.
That's my hope. The realization that in terms of, pardon the expression, but disease burden, there are many more individuals affected by this than I think are diagnosed, and witness many more girls that we could, and boys as well, but especially because this is an excellent condition, girls, individuals that we could be helping.
Yeah.
Probably not so much. Yeah, you can pause it. I think there's in the top right corner. Yeah, I know. It's frustrating. It's trying to.
So that's surprising because 1 in 1,500. We can do the math to see how many. So where are all these kids?
I think I calculated about 100,000 in the U.S.
So why don't we know about them? Because Rett syndrome is not a subtle disease, right? It's a serious disorder. These kids are going to neurologists. So so she's coming up with that number based on screening.
The Guardian study.
Yeah.
So they screened 10,000 newborns, and based on that screening,
okay,
which is still well, actually they've done 13,000 now. It's based on 13,000. So still a small sample size, but it's the first time that they've been able to get any kind of.
So I'm wondering, and so she knows the exact mutations of those. Yeah, it'd be interesting to see what the
She's asked me to pass on that you need to contact her.
Yes, I'm going to contact her today.
Yeah.
Because I'm wondering, so, some of these kids, maybe they have mutations that are going to be very subtle, there's probably that, so maybe these are kids that have mental health issues, will grow up to be, mental health issues, not full-blown. I would think if you have full-blown Rett.
You would know?
Well, I mean, look, if you're living in, Tennessee, Mississippi, in the rural areas, don't have access to a medical center. Yeah, we those kids may never get diagnosed, who knows. But, I mean, that's a big difference, 1,500 to 10,000.
I can tell, yeah. So I can tell you from our experience in getting Evie diagnosed, well, number one, it took 6 years.
Yeah.
Well, it took 6 years till we got a diagnosis. We didn't really start looking for till about 18 months.
Yeah.
She was falling behind, and we started to research. So I guess 4.5 roughly years of proactively looking.
Yeah.
And, I mean, this is back in 2016 when we finally got the diagnosis. Very different than now. But had we not continued to push and push and push and push, we wouldn't have gotten a diagnosis, and we would just be living with it today, which is, I think, what most families do. Because we have the means and we have the kind of education and all those kind of things to keep fighting, keep looking.
Yeah.
But I think most families don't have that. And they get to a point where they just don't have time, and it's just not even offered to them. Hey, why not get a full genetic panel?
Yeah. I mean, that number, that will change the the business model for companies that are pursuing not just, any kind of treatment and genetic medicines. That's, you're looking at a lot more patients. That's really interesting. Yeah, I'll contact her today.
Yeah. I was, when I heard that, I was,
I'd love to see that data, see what the mutations are. And are they, I'm assuming these kids are, anonymized, but it'd be interesting to track them, to see, are they all going to have symptoms? Are there some?
Well, I'm sure you could probably track them kind of backwards. I mean, they just got a Rett syndrome diagnosis, so they're probably going to be reaching out to the foundation.
Well, no, if it's screening, they just have blood samples, right? So, or saliva.
I think they do this through blood spots.
Yeah. So they probably don't know who the kids are. So we don't know if the kids are diagnosed clinically. They may have a mutation.
But I think they would have reported this to them.
Would they?
Yeah. It's so basically, the whole purpose of that study is for early screening, early diagnosis.
All right, so
they would have been diagnosed.
And these are all are they all kind of all New York?
Yeah.
Wow.
Yeah.
Okay, and they're all
babies.
They're all
newborn
babies. Yeah. Yeah.
So the families have gotten the diagnosis.
Yeah. Okay.
And the families will probably reaching out, to the Rett Syndrome Foundation.
There's a clinic at Montefiore in the Bronx, a Rett Syndrome clinic.
Mm-hmm.
Yeah. All right. I'm eager to learn more.
Yeah. No, it's the whole everything.
Now, what did you say about other disorders? Were there other disorders that turned out to be a lot more prevalent? Do you?
I don't know.
Okay.
I don't know. We didn't talk about other disorders. We kind of focused on Rett because of this. Phelan-McDermid syndrome wasn't even on the panel.
Okay.
They basically had two panel. They had the first series of things they were looking for. Then they added a bunch of other ones. But Phelan-McDermid syndrome was not on there.
Okay.
I think they still only I think the Guardian study, I think they didn't look at everything, right? They looked for, I think, 400 or something like that, particular genes.
Okay. It's interesting.
And then there's, we can talk about it later in terms of the early diagnosis and kind of what your thoughts about on that are. But let's get back to.
Okay.
Let's get back.
Thank you for sharing that.
Yeah. No, I was very excited, and Wendy was very excited.
Well, this is what we need. We need this kind of just screening that's not that's more comprehensive.
Mm-hmm.
Yeah.
So, 25 years ago, it's been since you've started the Rett Syndrome Research Foundation.
Yeah.
And then I think it was like 7 or 8 years later, it wasn't the foundation that turned into the trust. The foundation kind of continued and you started the trust.
Yeah, the foundation, the Rett Syndrome Research Foundation, merged with the original organization and became the International Rett Syndrome Foundation. I really wanted to focus exclusively on genetic medicines with no distractions, and I started the Rett Syndrome Research Trust.
So that's been going for what, about 18 years now, I guess, roughly?
So I started in 2008. So what is that?
16 years? 2008? Yeah, 16 years.
Don't show how bad I am at math. Mental math.
I cannot imagine that you are bad at math.
No, I actually like math, I was good at it, but I'm very bad at
quick mental math.
Yes.
Yeah, I think me too. When somebody's asking me something, it's if I do it on my own, I kind of move quickly. But as soon as there are eyeballs or somebody waiting for an answer, I just I collapse under the pressure.
But it's 16 years.
So looking back at those past 16 years, what are the big moments that kind of jump out at you?
Well, I mean, if I look back at, the whole 25 years, the big moments are, the discovery of MECP2 being a cause of Rett syndrome, right? That happened just as I was starting the foundation. So my first foundation. So it was, the timing was really great because it was a great time to jump in because, we had something to sink our teeth into, right? Mm-hmm. We know the gene now. I think, the first animal model was made, getting people to work on it, getting some recognition around the disorder was important. I think the next big important landmark moment was 2007 with Adrian Bird's Reversibility paper. Mm-hmm. That really changed everything. I think after that was funding our first gene therapy projects, which started in 2010, starting to get some intriguing preliminary data in 2013. That was Gail Mandel and Brian Caspar and Adrian Bird together, joint paper. And then we formed a gene therapy consortium from 2014 to 2017, 4 labs working collaboratively. And then it was data from that consortium that incubated and generated the data that led to Taysha and Neurogene licensing those programs and moving them along with their own funding. So I think, the first company that started a gene therapy program was Avexis. They got purchased by Novartis, and then Novartis terminated the Rett program. So that was a low point. Mm-hmm. A big disappointment for me and for the community. Fortunately, we had other irons in the fire, and this is a really important concept, right? Mm-hmm. You can't put all your eggs in one basket. Oh, I'm sorry.
That's okay.
You can't put all of your eggs in one basket or one modality.
Yeah.
It's just too risky. There's science risk, there's business risk, and you have to mitigate that risk by, in parallel going after multiple approaches, working with multiple investigators. That's really important.
Did you have that viewpoint before they
I did.
Got out, or and that just kind of hammered it home?
Yes, I had that concept before, and thank goodness, because there were other programs that we had funded and that were moving forward. So. But I think it probably hammered it home for the community. It's like, let's not put all of our eggs in one basket. So I think, getting companies involved and kind of validating that, yes, Rett syndrome is a disease that we're interested in, that we think would make a business model, that we can, those were pivotal moments. And, then the gene therapy trial starting is exciting, and we're waiting now for some data. Neurogene is going to share some data by the end of the year. Taysha is going to share data first half next year, 2025. And those will start to give some clues, about how well they're going to work. And also, the genetic medicines are made up of two components: what you're trying to deliver into cells, but also the delivery technology that is being used to get those cargoes into cells. And for now, the gold standard is AAV9. How well, that's a viral vector, right? How well AAV9 will work for Rett syndrome, we don't know yet. And so these first two gene therapy trials are going to give us some clues. And we may end up needing something better than AAV9. I don't know. Or hopefully it'll, we'll get some good results, but we suspect there'll be room for additional improvement.
I've now just come in contact with, do you know Paula Evans at
Yes.
Angelman syndrome? And I've kind of, similar kind of general, story. And I can only imagine all the other kind of future Monikas and Paulas who are getting a diagnosis, and it's going to be more and more and more as genetic screening becomes more popular. What kind of advice would you give to somebody who is, just gotten a diagnosis for their child, and there's nothing out there?
Yep.
And they will have that same, I've got to find a cure for my kid.
Yes. Well, I speak to those types of parents a lot because I get a lot of emails from parents that are seeking advice, and I'm happy to give it because I got a lot of good advice and a lot of mentoring from a lot of people, and I still do. So I'm happy to kind of pay it forward. So at least once a week, I'm talking to somebody who's, just got a diagnosis and wondering what to do, who wants to kind of follow in our footsteps. Well, what I'm, my advice is, first of all, do it. And it took, obviously it took us a long time, but science isn't linear, and you're not starting from scratch necessarily, because there's kind of a roadmap out there. And invest in kind of building some foundational building blocks that will get companies interested. So for example, patient data through registries, digital natural history studies, biorepository of patients' cell lines, mouse models. So these are kind of the building blocks that companies are going to say, do you have this? Do you have that? Do you have this? And the more that you can check off, the more likely they may be to become involved in your disorder. And there's ways now that you can, for example, patient data. You don't have to do traditional natural history studies where if you need to collect 10 years worth of data, guess what? That's going to take you 10 years. Yeah. Now you can do digital natural history studies with medical records, for example, where you're, collecting medical records, and you can, machine learning, extract information. So there's cheaper ways of doing it. There's registries that are already, kind of plug and play. Don't have to build one from scratch. So there's a lot of good stuff out there. And I think, typically these families are going to have limited funding. There are not a lot of patients out there. There's very little money. So you've got to use whatever money you have very efficiently. Then I say try to hire somebody with industry experience, if not full-time, part-time, a consultant, or, maybe you can share it among several new foundations. Try to get somebody with industry experience, because, if we don't have that experience, and it's really critical. And, right off the bat, think about, are you going to fund a lot of basic science? Do we need to know a lot? Or can you kind of skip ahead and start doing some experiments, just can you find somebody to take that gene, put it in a vector, deliver it, see what happens, So those are the types of conversations and strategies that need to be discussed. But I also tell them it's if they can do it full time, if they have the ability, maybe one spouse is working, one spouse can, it's a huge effort, and it's very difficult to get anywhere if you're only going to be doing it a couple of hours at night and on the weekends. So not to underestimate what it's going to take.
Right. Basically, it takes total.
Yeah. And the other thing is try to find as many families as possible, because the funds typically come from the efforts of families who reach out to their networks. And if you don't have any families, and if you're not independently wealthy, you are going to need funds.
Yeah. Yeah. And you guys have raised an impressive number of funds over the years. So how much is Rett Syndrome Research Trust Group?
We've raised $100 million.
Over 16 years?
Over 16 years, yes. Yes, and I can't take a lot of credit for that. It's really the families that work with us who refuse to accept the status quo and who stand up and say, I'm going to help drive this research forward. And it's not easy asking for money, but they reach out to their networks year after year. And, it's a pretty small group of people, to be honest with you, that are raising the money. And money, I recently, we recently hosted a webinar to update our community on the state of our genetic medicine programs. And something that I said at the end of the webinar that I'll share with you, about 15 years ago, I went to a science meeting in Houston that was organized by Huda Zadbi, and there was academic scientists and industry scientists there. And it wasn't just on Rett, it was on neurodevelopmental disorders. So there was a lot of, people representing a lot of different fields of expertise. And I stood up at the end of the meeting and I asked a question: if Bill Gates's daughter had Rett syndrome, how further down the road would we be? And the answer was, not that much further. Like, we still have to, there's so much science we have to learn. And that takes time. And yes, money will make it go faster, but it's still going to take time. And, we couldn't do gene therapy to the brain back then, even if we wanted to. It wasn't a question of money, that just couldn't get things into the brain. And we're in a very different situation now. Now I think money is the biggest roadblock right now. We could be more aggressively pursuing every single approach with more money. We could be funding more delivery options. We could be getting more companies involved. So, money is a roadblock now.
Yeah, I think Guping said the same thing.
Did he?
Yeah, now it's not a question of if but when, and that when is a direct kind of correlation to how much money is put into this.
That's right.
The more money, the faster we'll get there.
Yep.
And
and that's not to minimize the scientific challenge. We don't have everything figured out. We don't have a perfect delivery to the brain. But money will have more of an impact now, I think, than 15 years ago.
Yeah. So in terms of, so where are we here? So I was looking through on your website, the board of trustees, and not just the board of trustees, but everybody at RSRT. And every time I'm reading one of these bios, my jaw just drops. The caliber of people that are part of the organization and are help leading it, and what I obviously noticed for just about everyone, they have a daughter or a granddaughter with Rett syndrome. So, I mean, in the past, the belief was that maintaining like a strict separation between like patient communities and medical to make sure there was unbiased information. And and that kind of made everything very kind of very cold, very clinical. How has RSRT kind of demonstrated that you need that close collaboration, that you need buy-in, that you need that contact to really push things forward?
Well, our board, I think almost everyone on our board has a family member. We Adrian Bird's on our board. He obviously doesn't, and we have one other board member that does not. But everybody else has a child, a sibling, or a grandchild. I think that's really important. They're the ones that are motivated to raise money. Mm-hmm. Our staff, our chief development officer has a child with Rett. The rest do not. So our chief technology officer, chief medical officer,
chief marketing officer, didn't she also? I thought or maybe I'm mistaken.
No, just our chief development officer. Of course I do, right? So I think it's important. Years ago, there was this sense that if you had a child with Rett, you just weren't professional enough, your emotions were going to, lead you astray. And I thought that was ridiculous then, and I think it's even more ridiculous now. I've been doing this for 25 years. I can tell you, I'm not just a parent. I've learned a heck of a lot over the last 25 years. Now, I'm not a scientist, but I don't need to be. I understand enough to know who to bring into the organization. Then you hire people that know a hell of a lot more than you do. You surround yourself with people that are smart and really knowledgeable. But I think having so many people that have a personal connection to the disorder motivates us, and it imbues the whole organization with urgency. Yeah. Right? Time is of the essence. Patients are waiting. Our children are waiting.
Yeah. No, I agree. I think that type of urgency is really only truly felt when you're.
I think also I was at a meeting years ago, and it was a workshop, and it was a workshop on negotiating contracts with MTA offices. And anyway, we're going around, and everyone was asked to make one statement. And one person said, "Never say anything in a meeting that you wouldn't say if there was a patient in the room or a patient family in the room." And I think that's kind of, that's a good thing to keep in mind. And we always have patient families at every one of our meetings, right? Because we have so many people that are affected by this disease at RSRT. So I think it's important.
Yeah, no, I agree. So Chelsea was diagnosed early. She was diagnosed at 2 years?
She was diagnosed clinically at 2, and then a year later the gene was found, and she had a genetic diagnosis.
And I from your website, I read how one of your trustees, Marcy, she had a daughter who was misdiagnosed at 3 and then diagnosed again at 17.
Yes.
The reason I wanted to talk about this is because of the conference I was just at.
Yeah.
How does early diagnosis help a family with Rett, with any kind of genetic disorder?
Yeah. So, first of all, with that particular example, it's because that child, now young woman, was higher functioning than most kids with Rett, and so no one thought that she could have Rett syndrome.
Okay.
But I think it's important to get a diagnosis as early as possible for a number of reasons. One is it gives families a community to be able to belong to, and to have answers so that they're not wondering, God, is it that glass of wine I had when I was pregnant? Was it that, cigarette I shouldn't have smoked? Was it that whatever, the fall that I had, or, whatever, a million reasons that we beat ourselves up as parents, right? And so I think knowing the cause, also helping to plan for, future pregnancies. Are you planning on having more children? And I think that's really important. I think for the child, I mean, children, whether they have a diagnosis or not, are probably getting therapies. Kids with Rett syndrome or these other, very serious debilitating neurodevelopmental disorders, whether they know what the child has or not, they're probably getting therapies.
Right.
But with something like gene therapy now, let's say that it becomes available and commercialized, the earlier you get it, the better. Yeah. So, eventually I think these gene therapies are going to be delivered maybe in utero or at birth, so that hopefully these children will never develop symptoms to begin with. So it's easier to stop a symptom from developing than to reverse, I suspect. So the earlier, the better.
Yeah. At the conference, really kind of across the board, the way that it seems like everyone's approaching this is only looking for
prenatal testing once there's a
treatment.
Yeah.
And so I got up and kind of made the point that that's great.
Yeah.
With the actionable treatment, I think that's what they were looking for. And I made the point that, what's actionable is different for parents than what's different than for clinicians. Yeah. What's actionable for parents is this entire conference that they were at, newborn screening, would not be there if it weren't for diagnosing things that didn't have a treatment.
Yeah.
And it wasn't until I said it that everyone in the auditorium was very quick to support, but nobody was talking about it. The entire conference, they were just kept on talking about presenting their findings after findings of only looking for the things that are treatable, like what's on the RUSP and on the newborn screening panel is, they're just adding things like, I think, SMA was just one of the more recent ones because of Zolgensma. But it just seemed the point they were trying to make, that from a public policy, from a public health perspective, they need to be able to treat it. And I was trying to make the point to them that there's so much more to it than just being able to treat it. There's everything that you just mentioned. Yeah. The community and needing the community to get to a point where you can raise funds and bring in.
Yeah. If you're identifying, if it's really 1,500 and not 10,000, think of all the extra families that could be contributing to the research, and belonging to a community. And also, you could be starting early intervention and therapies as a newborn instead of as a 2-year-old, which might make a difference. I don't know. But for some diseases, it will.
So I'm curious, for the Rett gene therapies that are currently going on, I think in one of your webinars and your Q&A, somebody had asked a question and you had made the comment that the therapies shouldn't start until after their regression. Why is that?
Well, that's not my opinion. That's So typically clinical trials for Rett syndrome. So typically clinical trials for Rett syndrome, for the age ranges for recruitment, they usually start at around 4. So after regression.
Okay.
I think with gene therapy, maybe they'll get lower eventually. I think one of the companies is now Taysha may be doing 3 years old. I forget the specifics. But, with something like gene therapy where you really want to intervene as early as possible, I think they can make the case about lowering the age ranges. But typically for small molecule drugs, they've started post-regression because they feel that so much is changing during the regression that it's difficult to actually know, what is it, the drug? Is it just the regression that's part of
stable?
Yeah, they want more of a homogeneous population and to actually be able to detect whether something had helped. And so they typically go after regression.
Oh, that's interesting. For Phelan-McDermid syndrome, the starting cohort is 2 to 9. But in that, so regression is part of Phelan-McDermid syndrome as well. Not everybody has regression, and I'm sure the same as in Rett. They don't. Those people don't suffer aggression or suffer greater aggression. It could just be because that person had more to lose.
Yes.
Right. So somebody who wasn't able to build up any of those skills may go through the same process. Is just not as obvious.
Yeah. Yeah. The one downside in Rett syndrome of an early diagnosis, like at newborn, is that families get that 1 year when everything seems to be normal and you're happy and you're enjoying your child, and you take that away, right? Yeah. When you tell them that your child is going to have this horrible disorder, and then they're going to be watching every day, anxiously waiting for regressions and problems, and so you take that happy year away. So that's the one downside. And if there isn't anything to give them in terms of a therapeutic, it makes it even more difficult. So we do have to kind of balance that. I think it's a tough one.
Yeah. No, it's. I think that's one of the worries from the community, is that you're giving parents information that they can't do anything with. Well, it's not that they can't do anything with, but
it's bad news. It's terrible news.
Yeah.
Yeah.
Yeah. It's a tough call. So the current, let's talk about the current trial. So we've got there are two, just Taysha and Neurogene, or is there? Okay. So Taysha use AAV9 through ICV.
No, Taysha is intrathecal.
Oh, intrathecal. Oh, Neurogene is ICV.
Yes.
Okay. Sorry, I got it mixed up. Intrathecal is through the spinal fluid.
Yes.
Okay, and ICV is into the brain directly.
It's into the ventricles of the brain. Yeah. Okay. One injection into the ventricle of the brain.
Are those either or both of those using gooping things labs technology?
No. So we're funding Guoping to develop base editors for Rett syndrome. So it's, in gene therapy, you're not fixing anything. You're not editing anything. You're adding extra copies of a gene to compensate for the mutated one. Then there's editing approaches where you're actually fixing either at the gene level, the DNA level, or the RNA level. You're actually fixing the mutation. You're not delivering extra of anything. You're fixing the mutation.
Is that fixing in the body or outside the body?
No, in the body, in the brain.
Yeah.
Actually fixing the mutation. So in for what Guoping is doing, which are base editors, you're actually developing a therapeutic. You'd have to develop it on a mutation by mutation basis. Not all mutations are going to be amenable to base editing. It's either point mutations that are due to a single nucleotide base, or nonsense mutations.
And for those point mutations, did I read correctly that it's not all point mutations?
It's not all point mutations. So my daughter's mutation, for example, is going to be difficult to base edit. And that's because of the nucleotide bases that are surrounding the mutation.
Okay.
Could lead to, off target. They're called bystander effects. So you really need to look at it mutation by mutation. Some are going to be easier to fix with base editing than others. But base editing is just one approach that we're pursuing. There's others as well.
Let's talk about those. So there's the package, and then there's the delivery. So for the package, let's I think this all kind of goes hand in hand with your new roadmap to Cures 2.0. So do you want to kind of take us through? I know you've done it a bunch of times online and everything, but you want to kind of talk about what the roadmap for the next 4 years looks like?
Sure. So earlier this summer, we announced our strategic research plan roadmap to Cures 2.0. In 2017 to 2020, we had our original roadmap to a cure. This one is plural, roadmap to cures. And in the original plan, our goal was to identify all the various ways that we can boost levels of MECP2 and to start programs funding those various modalities. And we did that successfully. It was a $33 million plan. With Roadmap to Cures, the goal is to advance those programs to select 3 and bring them to clinical trials by 2028. So there's a number of different modalities that we're pursuing. So gene therapy is already in the clinic. There's base editing, which is a mutation by mutation specific, and it only applies to a certain type of mutation. There's prime editing that I kind of describe like base editing on steroids because it can fix mutations that are physically close to each other on a gene. It's genome editing, so you're fixing mutations in genes. By the way, base editing and prime editing, neither one of them exists naturally in the body. These are modalities that were engineered by David Liu, scientist at Harvard. And so with prime editing, eventually you'll be able to fix any kind of mutation. So mutations that are like insertions or deletions where large chunks are missing or inserted, or mutations that are like point mutations, but that can't be base edited. All of those will be able to be prime edited. And you'll need a lower number of therapeutics because you'll be able to fix multiple mutations with one therapeutic. That's prime editing. Then we have RNA editing, which is kind of, the base editing counterpart, but in RNA instead of in DNA. But there's only a certain type of mutation that can be RNA edited, either nonsense mutations that end in X. Those are premature stop codons mutations, or G to A mutations. So a particular kind of nucleotide base, because that's the enzyme that has been identified that can catalyze and can, bring an A back to it, bring a G back to an A,
which is what my daughter has.
Okay, so that can be be RNA edited.
So that's where base editing currently would not work, right?
Now, base editing could still work for some of those. So you could have multiple modalities that work for a particular mutation. So base editing could still work for a G to A,
or is it A to G? There was anyway. I was looking into it, and I was looking to base editing, and the variant that my daughter has is not captured under the current base editing.
Okay,
I can't
maybe because of bystander mutations or bystander effects. So I could take a look at it and Guaping would be the perfect person to ask about that.
Yeah.
But so there's base editing, prime editing. We've talked about RNA editing. RNA editing, by the way, can be delivered with small RNAs. It doesn't need a virus.
Yeah.
So you could get multiple modalities, because with viral vectors, you can only do that one time.
Yeah.
And then we have an epigenetic approach where we're trying to reactivate the silent MECP2 on the inactive X chromosome. And then we have something called RNA trans-splicing, where we're actually trying to, so when proteins are made, the DNA makes a copy of itself, right? That becomes the RNA, which exits the nucleus and goes to the ribosome where proteins are built. And that RNA is initially made up of something called exons and introns. I kind of think of them like chapters in a book. But when the final protein gets made, you remove the introns and you splice the exons together.
Okay.
So in Rett syndrome, 97% of the mutations are in exons 3 and 4. So as that removing of the introns and splicing of the exons happen, we can try to drive the process so that we're removing the 2 exons that have 97% of the mutations and replacing them with healthy exons. So that's called RNA trans-splicing. This was a field that very few people were working on. There were no companies, no trials, but that's really changed in the last couple of years. There's now companies that have started. There's companies there's startups. There's academic scientists that are spinning out companies in this area. So it's kind of an exciting area that's coming into its own. So those are the approaches, all of the different modalities. And then on the delivery side, the gold standard for brain diseases right now is AAV9. And, AAV9 works fairly well. It can get into some parts of the brain and maybe transduce 10, 15, 20% of cells. In some parts of the brain, it's low, 3 to 4%. But then you've got parts of the brain, especially the deep brain structures, where AAV9 can't access that at all. And there are challenges with viral delivery. I think one of the biggest challenges is that you can only give it one time. If you give it again, your body will mount an immune response, which could be very severe or even deadly. And so, we have one shot at selecting what genetic medicine that's going to be delivered with AAV9 that we want our kids to have. There are companies that are working on trying to figure out a way to redose, and maybe they will be successful. The other challenge with viral delivery is that it's very expensive. So it cost to companies $1.5 million to make a dose of AAV. So very, very expensive. And that's why these genetic medicines are costing, $3 or $4 million. And then there's a lot of effort trying to identify non-viral delivery technologies. And so we're going to be investing. We have invested and we will be providing additional funding for non-viral delivery technologies as well. The hope is that somebody will make a breakthrough and we'll be able to, redose, and there won't be cargo size issues because you can put very large cargos in non-viral delivery, and they're cheap and easy to make.
And I guess it's very early on, but like when you look at these different modalities that you're funding, is there kind of anything that's kind of a little bit ahead in the race in terms of?
Well, gene therapy obviously is further ahead, right? It's been around for many years, although with recent successes and a lot of failures early on. But so that's the furthest, and then I think followed by base editing. Because, we can get editing efficiencies pretty high with base editing. Prime editing is, I think, a couple of years behind that, as is the trans-splicing and the reactivation. So I see this kind of as clinical trials and then hopefully approvals coming out in kind of phases, right? So at first there was just gene therapy. When RSRT started, there was only gene therapy. These other modalities didn't exist. Right. So then there was base editing, and then there was prime editing. And so the goal here is to fund programs in each of these areas. We don't know which one's going to work best. The ideal thing is will be to have a therapeutic that addresses all mutations. It would be difficult to have something where you need to go mutation by mutation, expensive and difficult. But we think we can do it. And part of it is the FDA starting to think about, how do we accommodate for a program like what Ping is doing with, you'll do the animal studies and all of your IND enabling studies with one mutation, pick that, and that's what you submit to the FDA. But then know that there will be these other mutations where all you have to do is swap out a guide, which is kind of your GPS, right? Instead of going to this mutation, you're going to go to that mutation, but everything else stays the same. So we'll need the FDA to work with investigators and companies so that they're not going to require all of these animal data and IND enabling studies to be done for every mutation. That's going to take too long, be too time consuming, and not practical, right? And so do your studies with one mutation, but then when you do your clinical trials, do an umbrella clinical trial, you'll actually be recruiting for all these other mutations as well. And so those are the kinds of conversations that I think need to happen and are happening with the FDA. And I think they will embrace this concept because I think it's the only way to, move these therapeutics forward that are mutation by mutation. You don't need that for something like trans-splicing or gene therapy or reactivation. Those should apply. Those are kind of generic. They should apply to any mutation.
I've been pleasantly surprised by people's collaboration with the FDA. I always have this idea of working with government as slow, red tape, bureaucratic, unhelpful. they're not there to help, they're there to get in the way almost. And the feedback I've received from everybody that the FDA is really, really helpful and really seems like they want to be helpful in kind of all these new modalities, and with the IND, with investigative new drug, and all the platforms they have for advancing these kind of rare.
So at the FDA, there's two main divisions, CBER and CDER, and gene therapies. Most of them fall under CBER, so the biologics division. Mm-hmm. And the head of that, Peter Marks, is, I think, a big champion of rare disease, and open-minded and creative, and trying to figure out what are the challenges and the roadblocks and the bottlenecks, and trying to address them. There's a pilot study, for example, that was recently launched that mirrors Operation Warp Speed that happened during COVID.
Right.
How do we break down barriers and get communication between industry and the FDA to happen quickly, in a more informal way, where you're not waiting months for a meeting, and months for feedback. And so they launched something, the acronym is START. I actually can't remember exactly what that stands for, but industry could apply to be one of the chosen pilot program, what's the word? To be chosen to be part of this pilot program. And Neurogene applied, and they were selected. So we'll see, but they're having informal, more frequent discussions and getting feedback, and that should help move things more quickly. Wow. And if it works, this is a pilot program. If it works, hopefully it'll be expanded and available to everyone.
So all the funding that Rett syndrome does, do you ever coordinate, because you're moving so many different modalities forward that are in kind of early research phase? Do you ever coordinate with other trusts in terms of, for example, like all the funding that you're doing for research for delivery mechanisms? Mm-hmm. That's benefits everybody.
Yeah.
So is there ever a thought of a coordination with other groups that are raising funds for research to make sure that this, different funding dollars aren't necessarily going to reproducing the same scientific research in different places, which has some benefits, I guess, as well. But at early stages, like you guys, you pick many different modalities. Would it make sense to work with other?
Yeah, there are areas where I think coordinating with other disease groups makes sense, and delivery, could be one of those areas. And there is some overlap between what we're doing and, for example, what Angelman's is doing.
Right.
We're funding some of the same people, and yes, so there are just certain patient advocacy groups that are very like-minded in terms of to us in terms of what we're doing, and we speak to them often, and have been speaking more recently about what can we do together.
Mm-hmm. You're a force of nature.
Thank you. Thank you.
No, thank you so much for taking the time to.
Oh, you're welcome.
This.
You're welcome.
I'd like to thank Monica for her amazing work and taking the time to speak with me. She's truly a force of nature. Gene therapy carries tremendous promise and real risk. Parents who enroll their children make brave, thoughtful decisions in the hope of a better future. When there's a serious side effect or a tragedy, it's devastating for families, communities, and for the teams doing this work. We owe deep gratitude to every family who participates. Their courage moves the field forward. Now, some updates on the Rett trials. The FDA just granted Taysha breakthrough therapy designation. Taysha's pivotal REVEAL study will enroll 15 girls ages 3 to 22 with a 6-month interim look. In Part A, all participants gained or regained at least one developmental milestone, and new supplemental analysis shows consistent multi-domain gains across communication, fine motor, gross motor, autonomic, with 22 milestones and 165 additional skills achieved across the first 10 patients. Neurogene's registrational Embolden study plans to begin dosing in Q4 2025 across 13 sites. NGN 401 is delivered through a one-time ICV injection into the brain's ventricle. Earlier data show multi-domain improvement, with the first 4 participants gaining 23 milestones and skills. Newly released primate results suggest ICV delivery achieves a 4-time higher transgene expression in key brain regions over lumbar intrathecal, with no additional liver-sparing benefit with intrathecal. Sadly, in November 2024, a participant in the high-dose cohort suffered a severe hyperinflammatory reaction and died. The FDA stopped the high-dose trial and allowed the low-dose trial to continue. We also wanted to send our love to the STX-BP1 community. The first child dosed in the Capsida CAP2 trial, administered through IV, died 2 days after being dosed. That trial has been put on pause while they investigate the tragic loss. Our immense thanks and love to all the families taking part in these trials and making progress possible. Thank you for joining us. For updates, please subscribe, share, and visit generationhope.co. Keep hope alive.